woke up this morning noticed a little bit more chill this morning in the air. Probably means Cafe Scientifique season is getting near the end. We are on a penultimate presentation this evening, but before I introduce our speaker, just a few announcements. First off, and I mentioned this last week, but on your seats are the green forms for feedback for this year's series from Cafe Scientifique. We really appreciate any feedback that you can give us. Either leave it in the envelope on your chair or put it in the box at the back, or if you're a real technology whiz, it's online now on our website, and you can follow the link directly from that home page from www.bigelow.org. And so we do appreciate any comments you have as we think about next year's program for Cafe Scientifique. Our final presentation next week will be a guest presentation from the director of the Mount Desert Island Biological Laboratory, Kevin Strange. Kevin arrived recently, well, about a year ago, up at MDIBL from Vanderbilt University, and he's going to talk about the worm in us all, what primitive ancestors tell us about human disease and aging. And Kevin is an internationally renowned physiologist. I think he's an excellent speaker, and it'd be great for another fellow non-profit institution here in Maine to be able to come down and share some of the Boots Bay hospitality in that final presentation of the series. But tonight, I'm delighted to introduce one of our own from Bigelow Laboratory, Dr. Nicole Poulton. Nicole is one of our research scientists. Her background really is a PhD scientist, is from the MIT Woods Hole Oceanographic Joint Program, and she is now part of the laboratory at Bigelow, working in the J.J. McIsaac facility for aquatic cytometry, and she'll be talking about the uses of flow cytometry in this presentation. Nicole is an active seagoing scientist. I hope you're going to tell us all about what it's like to try to control one's bodily functions at the same time running a flow cytometer, because she has done some of this work in some of the world's toughest oceans, particularly the North Atlantic, and I can speak from experience because I've been in that patch just down there. In fact, just a slight anecdote, the world's largest wave ever recorded on an oceanographic research vessel was just about here, 132 feet high. The vessel was 85 foot long. That's fairly scary. Nicole, as well as undertaking this kind of work, has also been part of the International Tara expedition, which is a circumnavigation of the world, this time by sailing vessel, and collecting marine microbial data in a systematic way. For Bigelow, Nicole is highly involved in our education program, acting as the program coordinator for the Bloom program, which is our high school student program in the spring of each year, and also helping us mentoring REU, research experience for undergraduate students, which is just completed after 10 weeks this summer. So we're delighted to welcome Nicole to speak to you, as she really represents very much the coming face of oceanography.
Everybody hear me? Yes. Wonderful. Just clip in. Thank you, Graham, for a great introduction, and thank you all for coming here. I know it's towards the end of the season here in terms of our cafe size, and the weather has been sort of changing a little bit. I just want to introduce my topic for this evening. Basically, it's Sea Truth, Chasing the North Atlantic Spring Bloom. I'm going to be talking to you a little bit about the North Atlantic today. But before I do so, I want to emphasize the fact that I'm basically one member of a very, very, very large team of scientists that have been working on this project. So a lot of the data I'm going to be presenting to you today, or some of the pictures that I'm presenting to you today, come from a vast number of folks. And this was actually on board in the North Atlantic when we were on board the RV Nor, which is a Woods Hole ship. And there's, I think, a couple people in the audience that are a member of this team, but basically it ranges, it's an international team. We have people from Plymouth University in England, Germany. We have folks from as far as the West Coast in the United States as well. And over here in the backyard, over here in terms of the Darling Marine Center, which is the main campus or the marine campus there. And then I have a few local folks here at Bigelow that are also on this team. So I just wanted to make sure I acknowledge the team and that I'm one component of this. So I just wanted to give you a little bit of your bang for your buck here, in the sense that why am I speaking to you this evening, and why is, the North Atlantic so important? So basically, I just wanted to emphasize the fact, and you may have seen in the article, that basically the North Atlantic sequesters, or basically draws down about 25% of the carbon dioxide by the ocean. So basically, just in this portion of the world, we have this large drawdown. And basically part of this is the carbon sequestration from the storage is basically occurs because of phytoplankton bloom that occurs. And so I just have a NASA photograph here of ocean color. This is depicting phytoplankton in the surface water. And basically there's two images here where you can see that in the winter time, it's dark, there's not a lot of nutrients to the surface that have accumulated. And what you can see here is that there's not a lot of phytoplankton, basically, but this coloration here is indicative of phytoplankton. It's actually looking at chlorophyll fluorescence. And what we're looking here in the spring, you can see this is an intense bloom that's occurring off the coast here. And what it's suggesting is that these actual cells are actually transporting carbon, because they're photosynthesizing the carbon from the atmosphere and actually aggregating those as organisms and basically falling and basically radiating out and sequestering that carbon into the deep ocean. Basically, it's our carbon flux, and I'm going to be talking about this carbon flux quite a bit. So why the North Atlantic? So I gave you a little bit of that information already. I'm going to put it in terms of basically our climate and how basically global warming also affects the North Atlantic. This is a little outline of my talk today. And then I'm going to talk to you a little bit about the carbon cycle. I know over the course of the summer there's been quite a few talks talking about carbon. So some of this may be familiar to you already, but also to those that have not seen it. And then I'm also going to talk about the marine food web and basically how that relates to us. In a sense, like, how are this all connected? Why are we studying this? Why are we studying the North Atlantic? So I'm going to give you a little quick, brief introduction to oceanography here, to talk about this region of the North Atlantic right here. And some of you may have seen or heard about this conveyor belt of water that moves around the planet. And basically what it is basically a formation of water that sinks from the surface and then transports itself around the globe. And basically this is happening because of changes in temperature and salinity on the surface of on the planet. And basically what we have here, you can sort of see it here in the North Atlantic, my key study area. There's these two pieces or pools of water that's called deep water formation. And this is happening, and this is occurring because of basically the water is sinking here, just to give you a quick answer to the question here. And what's happening here is basically this Gulf Stream, which we all know about here, is transporting very warm, and in this case salty water due to evaporation to the higher latitudes. And then it's rapidly sinking because it's cooling, so it becomes very dense. It's very salty, dense water, some of the densest seawater that you can have. And what will happen is that will sink right here in the North Atlantic. And this whole conveyor belt of water, one of the major sources of the sinking process occurs here in the North Atlantic. So that transport of heat, basically, from the equator areas in here, basically due to sunlight, is basically what's causing the movement of the Gulf Stream up, and it's that transfer of heat that's basically moving the currents around in that North Atlantic area. So, why in the North Atlantic? Basically, this is, as I said, the deep water formation, and this is some place where you actually can see this high salinity that we're actually seeing up here. But basically what we're also seeing here is this CO2 flux. As I told you, the water is sinking here at this point due to the physical structure of the water. And what's happening here is this is showing you a scale in amount of carbon flux into the ocean. So basically negative numbers means it's going into the ocean, and basically positive or red numbers means it's coming out of the ocean. And what we're seeing here is that in the North Atlantic, because of this sinking basically that's happening of the water column here, is that basically it's taking carbon from the atmosphere and also sinking it to the deep ocean. Okay? And what's happening is that carbon dioxide is dissolving in surface water. There's a large transport. We heard 2 weeks ago from Dr. Barney Balch about carbon and how it's actually causing ocean acidification because it's dissolving in seawater and causing carbonic acid. So how does this change? How does this flux potentially change, or this movement of water into the deep ocean change due to changing climate? I know this isn't resolved very much, but what we can see, we understand that there's these increasing surface temperatures that are occurring across the planet, and basically, especially in the northern area right here, and this is also impacting the North Atlantic. So how does this affect the North Atlantic? So we have temperature increase, so the water is increasing in temperature, and then we also have this is something else that Barney Balch talked about a couple weeks ago. He was talking about the reduction of Arctic sea ice. And this chart here shows you basically starting in 1980, moving to 1990 to 2000 to 2010. And what we're actually seeing here is this reduction in our ice cap due to these warming climates, so the warming climate. And because of that, the thickness of the ice is also changing. And then also, the amount of the ice sheet is actually reducing in size. And maybe within our lifetime, we may actually see the existence of no polar cap in the summertime. So what we're seeing here is that the salinity is changing again. So if you think about it, there's basically a reduction in the surface salinity. So I talked to you about that deep water formation. So what's happening is that potentially that flux to the deep ocean will be changing because we're changing the salinity in the area. We're also changing the temperature in that area. So again, we're talking about a carbon sink that's occurring here in the North Atlantic, and how that's basically moving out of the atmosphere into the deep ocean. So that's why we're choosing this area to study. So the carbon cycle, we're going to talk a little bit about that and how that relates to the North Atlantic and why it's so important. So this is basically looking at the carbon cycle on the planet as a whole. It's divided into two sections where we have the things that are happening on land over here, and we have the items that are happening in the ocean over here. And so basically what you're seeing is that a lot of the carbon basically is being actually taken up and actually being released by two processes. We have photosynthesis and respiration. We also have on this chart basically showing you where all the carbon stores are, and meaning where is carbon stored on the planet. So if you think about here in the soil, this is in gigatons. This is a fairly large number, okay? And the arrows represent gigatons of carbon per year and how they're transported from either the atmosphere into the ocean or from the atmosphere into actually plant biomass, for example. And so plants are actually taking up CO2, as we know, but so is the ocean. And so here, the number for photosynthesis on land is 120, but the gas-sea exchange here is 90. And a part of that is actually phytoplankton and the actual organisms that are in the surface called the biological pump. It's basically photosynthesizing and respiring, and in a sense is also transporting and actually taking up a net 2 gigatons of carbon per year. But, as we know, we're playing around with this actual equilibrium that we have with photosynthesis and respiration. And so by doing so, we have this issue that I'm going to be bringing up with regard to fossil fuels and how we're basically changing the equation a little bit in the atmosphere, where we're ending up with a net annual increase of about 4 gigatons of carbon per year in the actual atmosphere. The plants on land are doing a fairly good job of absorbing that carbon, But so is the ocean, and the ocean is actually also a key component of actually taking out that excess carbon. And again, we heard earlier this summer basically about ocean acidification and potential problems that could also be caused by just the dissolution of CO2 into the ocean over time. But what I'm also going to be talking about is the importance of this net ocean uptake, the flux to the deep ocean of carbon, storing it. Now, have you noticed the biggest number in terms of the carbon stores on this chart? It's sitting right here in the deep ocean. You have to remember, it's about 70, 75, 71% of the ocean, of the Earth is covered by the ocean. It's about, on average, 3 miles deep. So if you think about it, in the deep ocean, there's 37,000 gigatons of carbon stored in that ocean, okay? There's a fossil pool of carbon on land, but it's huge in the ocean. And there's even sediments in the deep sea that are also storing that carbon. So the residence times of carbon, this is very important in understanding how we're changing our climate. So again, I have two sides to this chart. Oops, going backwards here. We have the terrestrial side and the oceanic side over here. And basically what we're seeing here is the movement of carbon via photosynthesis into grasses, trees, and then basically it's being recycled into the atmosphere through time based on its residence time on the planet. For example, here grasses and trees would be 2 to 5 years until they die and then decompose and then regenerate their carbon. And photosynthesis actually, for phytoplankton in the ocean can be very short time scale. We're talking about days to years in some senses in terms of recycling of that carbon. But a lot of that carbon, as it ends up in the deep ocean, basically takes 100 to 1,000 years to be recycled. And the same thing happens with trees or things in the soil if it starts to get buried, for example. And it's not until it reaches the actual benthic area of the ocean or is buried in the soil and actually becomes very buried that we end up with residence times of carbon of being on the order of 50 to 100 million years long, okay? And the same with and this is where basically our fossil fuels originate from, okay? From this very old stored carbon, originating originally from things like trees and actually phytoplankton. So what I'm going to talk about a little bit here is I have a little schematic of the increase in CO2. And I just wanted to demonstrate it because it does give you a really good sense of how the Earth functions in terms of its CO2. And so I'm just going to start it here. It's a little bit long, so I'm going to cut it off at certain points, but I want to get it started here so you can actually see this. This is let me just describe this chart here as we're talking here. So this over here is basically the increase in CO2 starting back in 1979. So you can actually see it. The reason why I wanted to show you this is because this is mapping out through time, and it's actually seen here. This is the year date here. For some reason it keeps stopping, but I'll keep it starting here a second. And as this clock spins, this is increasing. And what you'll notice basically, here in 1979, we were down here at around 336 parts per billion of carbon dioxide in the atmosphere. And as we move, here's 1989, it's around 350. And then as we move upwards through this, this is the Mauna Loa record here. I think it's because I'm pressing the actual laser pointer that's stopping. So I'll just talk about it first and then but here's Mauna Loa here in Hawaii, and this is the longest continuous record of CO2 on the planet. And what we're seeing here is it's gradually increasing as the years tick by, and these are different other stations where other observations of CO2 have been made. And then here what we see is the equator, basically looking at the North Pole and the South Pole, and basically looking at this actual record over time. So what you're seeing here is the actual breathing of the planet over the seasons, and I'll show you this here. I can make it work. So without pressing the button, I'm just going to let this flow a little bit. You'll notice that up here in the North Atlantic, in the Northern Hemisphere, what we're seeing here is that since we have the most land mass in the Northern Hemisphere, this is where we're going to see the most exchange in CO2. So what you're seeing here is the up and down oscillations as the years tick by. In the wintertime, there's less uptake of CO2. In the summertime, there's more uptake of CO2 because of the biomass of plants that occurred. As we know, the leaves come out in the spring, for example. So what you're actually seeing here is the world breathing, in a sense. That's how I like to think of it, you know. But as it's breathing, we're noticing something else, that the CO2 is actually increasing on the planet. So what we're seeing here is that we're going to end up getting to the top of this chart. It's around 2006 right now. I'm going to now move backwards in time a little bit. So this is the current record that we have. This originally started back actually in 1958 or 59 actually at Scripps. And actually that's what this record is here. This was the actual NOAA site, the red lines. The green lines that are showing there is now we're moving back to around 1959. And basically what's happening is that this is the actual amount back then. It's around 320 parts per million. Now what I'm adding on here is ice core data. We're moving to a very young ice core, and this is giving us the change in carbon dioxide over the Industrial Revolution period, where basically onset. And then basically it's giving us right around 1800 the pre-industrial level of CO2 on the planet, which was around 278 parts per million. So we're going to continue. So what's going to happen now is that we're going to move to another ice core. So you can actually show, now remember, this is where we are currently, actually it's 2009. We're around 392 parts per million as of July, or last month. This is an older ice core, and now we're moving back through time. And now, again, what we're seeing here, this is before, basically this is 20,000 years ago, 40,000 years ago. We're moving back through the ice core through time, looking at the CO2 levels in the ice, trapped in the air bubbles within the ice. And what we're doing now is that even an older ice core will now actually show back to 800,000 years ago. And what it's basically, the whole diagram that this is showing is basically, here's the ice ages, the range in between the pre-industrial and the ice age here. Basically, the ice ages on average are about 185 parts per million CO2 level. And this is the oscillating back and forth between interglacial periods. So around the maximum, interglacial maximum that we saw was around 300 parts per million. So what we're seeing here, that we're basically skyrocketing in terms of our CO2 in the atmosphere. So I'm going to talk to you about why that is in a moment, or later in the talk. So we talked about the carbon cycle, and now what we're going to do is talk about the North Atlantic and put it in terms of marine food webs. All right. So the North Atlantic, I'm just going to talk to you a little bit about what's actually happening in the North Atlantic in terms of phytoplankton. So what we have here, in the wintertime, there's a lot of mixing that's going on in the surface layer, and there's not a lot of what we call stabilization of the water masses, or there's no stratification that's occurring. And that's due to the fact that there's little light, and that temperature has not started to increase as the spring climate changes. So what we're seeing here is that as the spring bloom occurs, as we see like on land, for example, when the trees bloom, for example, what we see here is that there's an increase in light, and also an increase in nutrients. And the process of photosynthesis occurs using a molecule called chlorophyll, basically, which is the light harvesting pigment that's in all plants, including phytoplankton. And this basically causes, as the light increases and the nutrients increase, and basically we have this stratification of the water column at the surface, basically allows a bloom to occur. And the bloom then feeds our food web, for example, our zooplankton, which will end up in the guts of fish, for example, which is then what we consume and what our fisheries are based off of. So who are the bloom formers? I'm a phytoplankton ecologist, so a lot of my role in this whole team and looking at the actual North Atlantic bloom is to actually look at the players that are involved in the North Atlantic bloom. And here are a few of them that we see. These are some of the primary players. They're the diatoms. The diatoms do not have the ability to swim, but they produce these very ornate glass houses made out of silica. And this is actually a pillbox shaped one here. These are also found in the North Atlantic. They're actually found globally, but, excuse me, in the Gulf of Maine, sorry. And we also have here Cattosaurus. This is a species that forms spines, so they basically become fairly large. But when I mean large, they're about the thickness of your hair, for example. So that's how small these organisms are, but they're being produced in a very enormous amount. As you can see here, a huge assemblage of these organisms when we're looking just at the North Atlantic diatom community. And a lot of my research was using specialized instrumentation on board the ships to actually look at these and actually to characterize and actually classify a lot of the organisms that were out during the North Atlantic bloom. Some of the other players that are out there are the dinoflagellates. These are not silica-based, but because of like within their name, basically they have flagella, and they actually swim through the water column. And what we're looking in the North Atlantic bloom is we're looking at the changes in the community structure over time, and basically looking at how things shift potentially from a diatom-based community to other types of communities that are out there, similar to like a meadow over time and how the plant communities change through time. So we're looking at these changes. But one of the most important things about the North Atlantic is this, again, this flux. There's this water that's basically sinking out, but it's also the sinking out, or the biological pump that's actually happening and actually moving carbon from the surface to the deep ocean, and by the process of photosynthesis. So here's that carbon that's basically being absorbed and utilized. Again, here's the photosynthetic equation. And here's the glucose molecule, the sugar that's being produced by light, or utilizing light. And we have here, what happens after the bloom occurs is that basically these organisms start to bloom in such large densities that they start to aggregate together. And once they start to aggregate together, they start to sink out of the water column, and they rain out literally like snow, but it's actually underwater. So we call it marine snow. What's also included in this are other pieces of organic material, such as zooplankton feed on this bloom, just like your predator-prey relationships that we see on land, zooplankton feed on them and are also contributing to this, and they're actually packaging up the phytoplankton in much tighter units, and it's actually causing it to fall out a lot faster in the upper ocean. And in this process, we have a lot of respiration, basically, that's going on. And remember, if you remember back at my carbon flux diagram, we saw that there's basically a positive exchange. There's photosynthesis and respiration that's occurring. In addition to that, there's also a much smaller assemblage of organisms, which I'm also looking at, such as bacteria and viruses that are also respiring CO2, giving off CO2, but also they're basically consuming these big, juicy hunks of phytoplankton that are falling down through the water column. And how much actually makes it to the deep ocean is very little, as I mentioned earlier, because a lot of it's being recycled and actually stored in that deep ocean water column as it's moving downward. So how do we monitor this bloom? It's a vast space, and as we know, as oceanographers are, we have access to oceanographic ships, similar to this one here. This is a vessel out of Iceland that we were using. But we can also use remote sensing. We can use satellites to look at these blooms, for example. As I showed you earlier, this is from a satellite from NASA. And we can also use autonomous vehicles. And this is what the key to my project, or the team project that we were on, where we were utilizing autonomous vehicles to analyze and look at the bloom. And so what we have here are, how do we monitor these blooms? Basically what we have, and we use floats and gliders. These are basically autonomous vehicles that basically can be deployed and have sensors on them that actually can monitor the water column when we're not there, for example, okay? So these are little, it looks like torpedoes, and basically this one's actually a float. It actually moves with the water. It's called water following, or Lagrangian. And then there's sea gliders. These ones actually you can control where they go and actually deploy them so that they're moving upwards and downwards through the water column. We actually put a series of sensors on these actual gliders and floats. We can look at conductivity, which tells us about salinity, temperature, and depth. We can look at O2 evolution. Photosynthesis produces oxygen, so we can look at how much oxygen is being produced in the atmosphere. We can also look at light transmission, chlorophyll fluorescence, and backscatter, which backscatter actually tells us how many particles are in the water column. So, in terms of gliders, this is just basically the schematic of it, or excuse me, actually this is a photograph of a glider sort of taken apart. There's a bladder here in the back of oil, and this is a battery that moves back and forth that allows it to move up and down. And it moves fairly slowly, okay? And I think I mentioned in my previous slide here that the nice thing about autonomous vehicles is that it can monitor continuously for a very long period of time and actually monitor what's going on out there when we're not there. So here, the range of these can be in the orders of almost 1,000 kilometers, and they have been deployed upwards of 7 months. On our cruises, because we had so much instrumentation on board and did battery life, we had an instrument out there for approximately 3 to 4 months. So this is a little schematic of where we were. This is Iceland again up here to give you, and this is Reykjavik, where we basically deployed from. What we did was to monitor the spring bloom. We deployed a whole series of floats and gliders to monitor the bloom over time, basically. And then what we went out to do is basically we periodically would go out to look and to see whether the gliders were collecting data properly. So this was in 2008. In early April, we deployed all of the floats and gliders, and then in May we did a large process cruise right during the bloom, where we could actually see the bloom structure. There were 4 cruises. Basically, the float, these guys were out there for about 70 days, and the gliders were out there for almost 340 days, combined across 4 different gliders. So this was not, so it's about 3 to 4 months, but divided it, there was 4 different gliders that were actually out there. And this was the track. We started off right here at this location where the star is almost, and basically, this is just due south of Iceland here at about 59 degrees north latitude. And then basically what's happening here is that this is the trajectory of the actual floats. It's moving with the water mass as it's spinning around in the water column. And the gliders are actually basically doing little bow ties around the floats, basically to look at the area around this patch of water that the float was looking at. So what can we get from this information using these autonomous vehicles? So this is what I call the anatomy of the North Atlantic bloom. So what we're seeing across the x-axis here is time. We're actually looking at the evolution of this bloom. And what we're seeing here is depth. And what you can see here is that the gliders, excuse me, this is actually the float data, but what we're actually seeing is that here is this sort of winter mixing. This is low chlorophyll levels here, the blue, and then high chlorophyll levels are the high. And again, chlorophyll is a proxy for phytoplankton. Okay, photosynthesis, all right? So what we're seeing here is that as time progresses, we're moving here, this is March to June, so around April, May, we're starting to see this increase in the surface layer. This is going down through the ocean here. So what we're seeing right at the surface here, later on, we see a stratification of the water. As the temperature starts to increase, the water basically stratifies right at the surface and allows this bloom to occur. The nutrients have built up there, and then basically we see this bloom using the gliders. We can actually see, using an oxygen sensor, the evolution of oxygen at this bloom. And then we know that phytoplankton require nutrients, and so we can actually see the decline of the nutrients in the water column using these gliders. And this is actually taken completely using glider data. We didn't actually have to go out and monitor this. The only thing is that we have to go and collect them. So we have to actually go out and put them in the water and take them out. Another thing we're looking at is talking to you about carbon flux in the North Atlantic. So this is data that's been put together by Nathan Briggs. I believe he's in the audience at this time. So this is looking at glider data, and what you're seeing here is backscatter. This is the particles in the water. As I told you, these blooms are basically comprised of these large phytoplankton flocks. And as time goes by here, and this is year day, so this is actually around April, and what's happening, this is the beginning of May, or end of April, beginning of May, and what's happening is that the number of particles in the water is increasing. So you're actually seeing that with the scale bar here, red meaning high, blue being low. And what's happening is that they're actually starting, these particles are sinking through the water column. And you can see this also in the chlorophyll. This is all those phytoplankton that are basically built up at that surface layer right there. And you can start to see and estimate the carbon flux that's actually happening in that North Atlantic patch, or that bloom that we're seeing. So this is the number we're trying to get at, how much carbon is out there. Ship calibration. So we take these sensors out there, and basically we have to understand, basically, are the sensors correct on the gliders, for example? So we have to actually go out. Now, Graham introduced you to the North Atlantic a little bit with high wave heights, and yes, it's one of the toughest places to work, actually, and the ships that I'm on usually experience the worst weather in the North Atlantic most of the time. It's not the best place to go in the spring, actually. But what we do is we actually go out to basically measure to see whether the actual sensors are correct. And so the way we do that is that we have to go to sea and actually analyze and look at the water column at the same time. And this is the RV Nor that we went out on. And basically we looked we basically performed these calibrations right next to the gliders or the floats right in the water. And basically what we're doing is we're actually physically measuring what the glider is sensing. So here's an actual photograph. We basically pilot the ship so that it's very close to the float. This is actually a float, and it's a small little flag. And believe me, it's amazing how you really cannot see these things until they're right up next to the ship. We have we know where they are based on GPS, but basically, other than that, you're looking for a needle in a haystack most of the time when we're seeing these things. We get as close as we can without hitting it, which is also rather challenging and very rough weather. And believe me, we have hit a few. And we've lost a few that way, but that's part of the problem with working in oceanography in a very turbulent system. So what we're doing now is that we're going to compare what the float is measuring, and we're going to measure it as well on board ship. And how do we do that? We use water sampling, basically, and this is a Niskin bottle, a series of Niskin bottles, actually, that basically collect the water at different depths. We send it over the side, we collect the samples, and then basically we look at fluorescence, backscatter, and we look at these different properties of the water, as well as the organisms in them, and actually then compare that to what the sensors and the gliders are seeing. And so my role on board ship was actually to look at the community composition, as I mentioned before. And to do that, we used a whole bunch of automated type particle counters. In this case, we have down here, this is a flow cam. It's basically a flow cytometer or microscope, which basically images the phytoplankton that are in the water column. This was actually an instrument that was originally developed at Bigelow and is now being produced by a company down in Yarmouth, Maine, called Fluid Imaging Technologies. We also have here, we basically to look at the zooplankton community, or to find out what they're doing, the players in that community. We actually had to deploy nets to actually collect the zooplankton and actually map out where they are. And then, of course, traditional microscopy to actually see who these organisms are. And then, in addition to the sensor information that we were getting, we also wanted to know physically how much carbon was actually starting to sink out from this bloom. So to get back to the carbon story, we had to basically, to determine the carbon flux, we had to put out something called a sediment trap. And these are actually water-following sediment traps where you can deploy them, and basically they go down to a set depth. And in this case, this one was set for 750 meters in the water, fairly deep. It's below the euphotic zone, but below the area where light can penetrate. And basically, it's capturing a column of water, basically over a certain period of time, and basically is determining how much material is flocculating out of the surface water. And so you can see here, beneath here, is basically there's these little cups, and there's these cones here on top. And they are set out for about 4 to 5 days at a time. And what you can do here is actually then capture this carbon. Okay, actually what it is actually phytoplankton. If you look at it under the microscope, it's actually phytoplankton that has sunk out of that surface water. And then from there, we can determine a carbon flux. We can actually measure that. So as I've already mentioned to you before, the surface ocean, if you think about the number of carbon atoms, if you take the equivalent of 100 carbon atoms at the surface in terms of the actual phytoplankton molecules, or phytoplankton particles, and as they drift through the upper ocean, basically it gets remineralized, it's being released, the CO2 is being released back into the water column, and into the deep ocean, basically at 1,000 meters, basically very little of this original carbon is making it all the way down. But one atom out of 1,000 atoms ends up making it to the deep ocean. But that's positive, at least it's moving towards, at least some of it is being sequestered to the deep ocean. So, why do we care? Why is this important to us? So we talked a little bit about CO2 earlier. So we know that it's increasing, and we know that it's increasing due to fossil fuel burning. And why is that a problem? Because what's happening on the surface of the planet on a normal basis, basically, is this cycling rapidly through the surface on terrestrial and in oceans. If you think about phytoplankton and how it's recycling over short spans of time, really. Basically, the amount of, only very little amount from the atmosphere is actually going all the way to the deep ocean and being stored in these really deep in these deep pools of carbon, for example. And these are typically have residence times of about 50 to 100 million years. And by the fact that we're burning these fuels, basically it's releasing very old carbon back into the atmosphere. So it's basically putting the carbon cycle a little bit out of balance. And so by continually burning these older fuels, basically we're bringing them up to the atmosphere and releasing them. And so this is the additive effect that we're having and that we're seeing in these 4 extra gigatons per year. And that's where we get this figure, basically. And these are the 4 extra gigatons that you're seeing. So, why do we care? And I talked to you a little bit about increasing temperature. We talked about increasing CO2, how the ocean is absorbing that CO2. But basically what you're seeing here is that there's these trends that we're beginning to see that as temperature, this is temperature anomaly, this is a change in temperature. And this is from 1997 to 2008. And where you see that there's a decrease in temperature, you see an increase in chlorophyll. This is from satellite data. And where you see an increase in temperature, you see a decrease in chlorophyll. So there's a pattern here that's developing. You can actually see wherever there's high temperatures, you see low chlorophyll. And so that increasing surface temperature, they believe, is being tied to chlorophyll, and that's what we're trying to look at when we're actually out here over time in the North Atlantic. So as carbon increases over the next century, basically, so if you think here, this is going into basically the future, if you want to think about it. So this is about 140 years down the road. And this is the CO2 as if we were to sort of stay on track as we are right now. I mean, obviously we'd like to see some reductions, but there's always, the population of people is increasing as well. So we're going to probably see this dramatically increase over time. So what we're seeing here is the concentration of CO2 increasing. And there's a correlation now that basically as the surface temperatures increase, and we know that CO2 is a greenhouse gas, and that's contributing to this problem of increasing atmosphere temperature, but probably also surface ocean temperature, we're going to see more of this stratification, meaning there's going to be less mixing that's going to be going on in the ocean. And we talked about potentially shutting down that conveyor belt of water. It's not going to shut down, but basically we might slow it down because we're changing how salinity, basically the properties of the water in terms of their salinity and their temperature. We're also going to see this increased stratification, meaning that because of less mixing, basically, there's going to be less nutrients that's going to stay in the upper water column, basically, and it's going to basically decrease the nutrient upwelling. And because of that, there may be a decline in these larger phytoplankton species, such as diatoms. And it's maybe promote more of a smaller phytoplankton community. But you have to remember that it's the large diatoms that are the ones that the aggregate formers. They're the ones that sink out of the water column. They're the ones that are going to be transporting that carbon to the deep ocean. So if you think about it, it's a little bit of a negative feedback loop. So in a sense, because we're increasing the CO2 in the atmosphere, we're changing how the ocean physically operates, which basically causes the biological pump of the ocean to change as well and its properties. So this, in a sense, by reducing the amount of large phytoplankton that's out there, there's a carbon flux reduction to the deep ocean potentially because of this. Now this is all, in a sense, hypothetical at this point. They're modeling it based on data that we currently have as the CO2 increases. But this is what we're going to be looking at, these changes in diatoms over time, or larger phytoplankton in general. And then, how is that altering the marine food web? Basically, if you think about it, especially larger trophic levels, such as ourselves, in terms of fisheries, for that matter. So as we basically move forward through this process, we're going to have to take a good hard look at this. And so again, it's all tracing back to why I'm in the North Atlantic, because I'm looking at these carbon fluxes through time, looking at changes through time, and monitoring this so that basically we can help basically put numbers into those basically predictive models like this so that to better estimate what's going to be happening in the future. So, and with that, I'd like to say thank you and see if there are any questions. Yes?
What exactly is the community shifting into as the larger diatoms and other planktons?
So a lot of what I study as a phytoplankton ecologist are some of these very small, tiny, what we call eukaryotic picoeukes. And so basically they're very small, they're about 2 to 3 microns in size. They are eukaryotic, meaning they're not bacteria, but they photosynthesize. But basically what we're seeing is that there's potentially a higher predominance of those over the larger phytoplankton, because they have a much, what we say, surface to volume ratio. Basically, they can absorb the smaller amount of nutrient that's available to them in that upper water layer. So in a sense, if we go to a more stratified ocean, basically what we're going to do is promote those over the larger phytoplankton. Yes?
You said that the phytoplankton start sticking together. Do they do this when they are alive or dead, or is there some combination?
It's a little bit of both. So the question was basically the sticking together of phytoplankton. Do they do this only when they're alive or when they're dead, or both? And what I like to say is when it's both, because what happens is that during a bloom, basically the nutrients eventually, for these larger phytoplankton, the nutrients eventually run out for them. And so what happens is that then they go into a sort of a nutrient-starved state. They're technically still alive, but what they're doing is that their properties are changing, and the bacteria are actually starting to attack them, for example. They're starting to break down, and they get more sticky when that happens. So what happens is that they start to clump, they glue together in a sense. They produce organic matter that makes them more sticky. And because of that, they start to sink down, and they clump together more in a sense, than they basically, there's more of a carbon flux in that sense. But it happens actually when they're still alive. And a picture I didn't show earlier, but they actually have different stages of their life cycle where they actually form a dormant stage at that point, where they don't need to grow, but they basically wait for basically spring again for the next season when they're mixed back up to the surface layer. And they almost hatch like an egg in a sense. That's what so. Yes?
As the CO2 increases over time, is there a practical way to put more nutrients in to increase the phytoplankton over time?
So the question was basically that we're having this increase in CO2, and is there any way, because of maybe the stratified surface layer, of adding nutrient to the ocean to produce, to grow more phytoplankton? Is that what you're thinking?
Yeah.
So there's been a lot of theories out there. There's actually a lot of nutrient-limited parts of the ocean, and there are areas where people have been hypothesizing, like adding iron to the ocean to cause these blooms to occur, to sequester more. But what we're finding is that when you stimulate a bloom, you also stimulate the other trophic levels. And so what happens is that how much of that carbon, if you were to add it, for example, would actually stimulate it enough so that you would get more than that 2 gigatons per year, that's a questionable debate. And then it's sort of like biological control at that point, and it's a little bit of a debate as to whether you can. And then also, to produce that amount of nutrient in large quantities and trying to put it into the ocean, it would be sort of like by adding, you may affect an area of like the size of a bathtub. So, so you need a lot more nutrient and it'd be very expensive. Yep.
Dr. Poulton, this may be a philosophical question, but this week we had yet another candidate for president deny the carbon cycle, deny CO2 issues or whatever. I'm wondering, how does science really convince the politicians that the science that they're bringing forth is really apolitical, but indeed a warning to us about the use of fossil fuels and all the other issues of global warming, the rising of sea levels, the issues of the phytoplankton. I'm wondering, how does science reconcile with politicians?
So the question is about science reconciling with politicians and how do we convince the politicians of what's happening. It is a philosophical question, and I do think a lot of it has to do with communication and actually communicating the results not only to the scientific community, because right now scientists do a good job of communicating with each other, but what we need to do is actually communicate that story in venues like this to a wider audience to try and substantiate what data is showing us. I mean, you can put political views aside to a certain extent, but also the data shows it, and it's in the data that we need to really communicate and actually have more venues like this in terms of actually being better communicators to the general audience, and it's really important in terms of communicating your science. So, because within the scientific community we understand, but outside the scientific community there's a lot of doubts, there's a lot of false data, there's a lot of, it's really trying to weed through all of that and trying to really basically inform the public. So that's how I'd say it. So, yeah.
Switching oceans for a moment, what's going on in the North Pacific, and does it matter? Or is it just downstream from all of these effects?
Well, we talked about that conveyor belt, and actually certain areas of the Pacific actually are areas where basically a lot of that deep water is coming back to the surface. And so there are high productivity areas, but it's also sometimes can be a flux out of carbon and out of the actual deep ocean as well. So there's other areas very similar to the North Atlantic. I think the North Atlantic is sort of a hot topic because it's such this large driver of because they have this large deep water formation that's occurring there. But there's lots of little hot spots all over the globe, including the Pacific as well. So, yes.
What basically goes on with stratification and with temperature increase? Can you summarize that?
Sure. So the question was, how do you describe stratification occurring of the surface water? So basically, the concept of stratification is that as you move through the seasons of the year, basically the upper ocean, basically in the wintertime, there's a sort of, it's based on like temperature and light and all those different properties basically allow basically the physical structure of the water is basically overturning in the wintertime. There's a lot more mixing, it's a rougher seas in that period of time. And then what happens in the springtime, there's basically a heating of the surface layer. So it's sort of like, if you think about it here like maybe in your backyard or if you're swimming out in the ocean, basically it'll form a warm lens of water across the surface. And so that's what we call stratification basically. It's this warm lens that's sort of appearing. And it can also be basically on top of nutrient-rich deep water, which was mixed up to the surface originally in the wintertime. But basically then the warming basically forms these layers to the water, and there's less mixing that's happening in those smaller layers. So what happens in those layers is basically the nutrients are being utilized to a certain point, and then they run out of nutrients because there's no mixing from the deeper zone. But once things start to sink out, if there's still enough light, you could potentially grow if you came out of that stratified layer. But usually that layer is basically indicative of where light penetrates, because that's where a lot of the heat is coming from. Anyone else? Yes?
Is the rise in the atmospheric CO2 accountable by the burning of fossil fuel? In other words, is it 4 gigatons of fossil fuels, carbon that is in the atmosphere?
It's not only the burning of fossil fuels, that is true.
But you said that clearance by sinking of carbon is about 2 gigatons.
Correct.
So is the overall process something that's also poisoning another mechanism of clearance that we don't know about?
Oh, in terms of the two that are going into the ocean, is that what you're asking?
Well, the rise can't be accounted for entirely by burning fossil fuels.
Correct.
So it must be coming from either another process that's generating CO2 or failure of a clearance mechanism.
Well, I talked to you a little bit about the negative feedback loops. So, like, as you increase the CO2, basically you're going to be changing those equations of photosynthesis and respiration basically are moving out of sync with one another. So what's happening is that there could be natural feedbacks from their own environment because of the anthropogenic sources of CO2. Basically feeding back and making a negative feedback loop, so it's just absorbing less CO2. So that increase could be due to the fact, the additional increases could be due to the fact that less CO2 is actually being taken up by the ocean by the biological pump, for example, because of the loss of more stratification, less larger diatoms, or larger phytoplankton, and so forth.
Do you know anything about how the actual ecology of the plankton in the ocean has changed since 1960? In other words, have the species changed substantially? Do you have data on that?
Well, in terms of species, I'm trying to say, what I'm noticing is not so much in terms of the types of species. The diversity still seems to be there to a certain extent in terms of the types of organisms we're seeing in the large phytoplankton. But what we're seeing is, what I've been able to see is that there's this shift to smaller phytoplankton. So there's just more of the smaller ones and less of the larger ones. That's what I'm seeing mostly. So, anything else? One more question over here.
So along those lines, I'm assuming this North Atlantic bloom is really good for all the fisheries around here in the North Atlantic. And then when you end up with smaller is that going to hurt the fisheries?
Yeah, so that's so the question was basically, if we move to smaller phytoplankton, will that eventually hurt our fisheries down the long run? So that's sort of a little bit of what my final point was about, is this marine food web is very important for us as in terms of like larger trophic organisms that are in the ocean. So if you move from basically a large phytoplankton dominated community to a smaller phytoplankton dominated community, it's going to change that trophic structure down the road in a sense. So basically smaller zooplankton will feed on them. So in a sense, there won't be as much carbon moving up through the food chain to feed and support this larger fishery that we're seeing. So in a sense, there will be a downward, it's sort of like a lower fish catch in a sense down the road potentially. So.
Nicole, thank you very much for illuminating really. One final presentation next Tuesday. Kevin Strange from Mount Desert Island Biology Laboratory. Thank you very much for coming.